lab number 12, and we'll talk about the flatworms, the mollusks, the segmented worms, and the roundworms. I'm still debating, do I need to keep that particular nematode, the roundworms, in this lab or move it to the insects, but insects or arthropods is a relatively large lab. But essentially remember that those ones are Lochatrophozoa and those ones Excitozoa, so they are slightly different. Very interesting group from the point of view of evolution, Platyhelminthes, Asalamates. Then we have a Nematoda, Pseudosalamates, mollusks, and Annelida fully have the xylem fully body cavity, so true salamates. Then we're talking about incomplete digestive system on one opening. The rest of them, well, nematodes actually have a full digestive, the tube, the mouth, and the anus. We're talking about all of them. Well, those guys are protostomes. So here we have Lochatrophozoa as well as the Excitozoa, so for phylum nematodes as well as the arthropods. So how can you tell the difference between female and male ascaris? Female and male ascaris are the roundworms and their parasites. I think I showed you those nasty pictures when you see the digestive tract and a bunch of worms sticking out. This is basically one of the common human, I'm not saying we have it, but that's one of the most common human roundworms parasites. The female will be larger, and you will see we have some of the pictures there, but first of all, it's larger, thicker, because she's supposed to take care of the eggs or produce tons of the eggs. But the main important, this is how you can tell male versus female. The male on the end will have a hook. Why exactly so? Tail is hooked. So why exactly it has a hook? Remember, they are located in the, well, they live in the intestines. It's very slippery in the intestines. All of the partially digested food, all the mucus, etc. So the food can go through the digestive tract without much of the problem, right? So in this particular case, whenever they're copulating, the male literally hooking up with the female. So this is how can you say, well, how can you tell whenever you're looking at the male versus female, you'll see the worm with the hook at the end, that's the male. So male has hook at the end. Name Mollusca class or classes which provide food for human consumption. Gastropods, right? We were talking about three major ones. Gastropods, snails, French cuisine right there. Good prepared snails are actually pretty good with the white wine sauce or anything like that. Yeah, that's good. Now, we talk about the bivalves, right? Oyster, Rockefeller oysters, or oyster Rockefeller, right? Or the baked oysters with the cheese, a little bit of mayo or aioli, and a little bit of garlic. Yummy. And then, of course, the calamari. Anybody tried calamari before? Here we go. That's the cephalopods, squids, essentially, right? Or so we have all that particular. So gastropods, bivalves, cephalopods. We eating pretty much all of them. Ascaris belong to which phylum? They belong to Nematoda, roundworms. Define protostomes. Protostomes are a group of organisms where during the embryonic development, the blastopore, the initial opening in the blastula, whenever the gastrulation occurs, will form mouth. Protostome, mouth will be formed out of the blastopore. Which phylum must shed to replace the exoskeleton to continue growth? There is a two phylum, arthropods, the insects, and we just talked about that. Well, insect is a class, but it's belong to the phylum arthropod. But also, don't forget the nematodes, the roundworms, the ascaris. They also shedding their shell whenever they're growing. It's not as strong as in arthropods. It's much, much thinner, but still they have to shed. All right. What organism can cause pain as the larvae form migrates through the muscle tissue? In this particular case, that's the worm, and we'll talk about that. That's a trichinella. Trichinella will cause trichinosis. That's called the trichinosis, and trichinella. The spelling is where is that? Right here. There we go. That's the trichinella. That's the worm, which is located in the muscles. Well, it's embedded, encased in the muscles. All right, earthworm belong to the phylum of Annelida. Members of the phyla exhibit the symmetry. They're all bilateral symmetrical. What is the importance of murex historically? Murex is the gastropod. It's a mollusk. It's basically a huge sea snail, but it contains a very interesting dye. It's a purple dye. But because murexes, you have to dive very deep. In the ancient times, we're talking about Roman times, ancient Rome times, each murex will produce just a minuscule amount of that particular dye. It was really strong, very beautiful purple dye. As a result, in nobility, very, very wealthy people were able to make their robes into a purple color. The same type of nobility or royal purple from the France. We have Louisiana is next to us, right? They have, well, I think the official color is purple. That particular purple color comes from that the dye comes from this particular snail, essentially the gastropod. All right, well, let's kind of take a look at the table number 1. Identify and sketch the planaria live on a display. This is the model of the planaria. Let me try to find the live one. Well, that's not the live one. Oh yeah, we don't have the live one, so you can sketch, well, that's technically a preserved one, but this is how the live planaria without any coloring will look like. Tiny little bitty creature, about quarter of an inch, a little bit longer than a quarter of an inch, somewhere between quarter to half an inch. And you can see the eyes, right? They're not image forming, they just to detect the light. And those ears, they called auricles, and we'll talk about those. So basically all you need to do is take the picture, snippet this particular image. So now whenever you're looking at the model, in the model you will see quite a few interesting things. First of all, you'll see definitely a head. You can, with the concentration of the sensors, as a result, concentration of the nervous tissue. So they essentially will have the brain. So 2 of those are eye spots. Now auricle, ears. It's not really ears, more they use to sense the environment. They have some chemical sensors as well as the touch sensors. But in this particular case, they look like ears sticking out on the side of the head. So see right there, that's the auricles. Now pharynx. Pharynx is the muscular organ kind of in the middle of the body of the actual planarium. And what it does, it's basically a sucking tube. It sucks stuff in, and then it goes into a digestive system. You can see that particular digestive system, very intricate. Remember, the circulatory system. So digestive system is actually taking the nutrients and delivering it to every single part of the body. As a matter of fact, in the video, which I sent the link to on the flatworms, it shows how those creatures are behaving relatively nicely. Now, gastrovascular cavity, all that particular red stuff is the cavity where the food goes being digested, and then whatever is not digested out from the pharynx again. So in this particular case, this part right here, kind of a tubular organ coming out from the center of the body to eat any kind of, or grab any kind of food. And then, of course, on this particular slide, you can clearly see the darker gastrovascular cavity. And again, on this, that's a slide, colored slide, you'll see the body, you'll see gastrovascular cavity, you can see the pharynx as well as the eyes or eye spots and the auricles. And this is what you're looking at. You'll need to say eye spot, auricle, pharynx, gastrovascular cavity. I just showed you. And this one is a side by side. This is how the planaria look like under the microscope. This is a colored planaria with filled up the gastrovascular cavity, so you can see some of the details of the body a little bit better. And that's a free living creature, quite interesting, very simple, not very well, not harmful at all in creeks around here. So anyway, now we're moving to the parasites, and the first group of the parasites were the liver flukes. CDC shows you a fairly complex cycle. Again, remember the eggs coming out have to end up in the water. The egg will hatch into the larvae. Larvae will have to get into the, and again, there is several stages of the larvae, but it have to go through the development into the temporary host or secondary host, usually a snail. The snail either have to be eaten by another temporary host, or the larvae will get into free swimming circasia, which will find the proper fish and embedded in the muscle of the fish. And that's one of the reasons why I told you the eating, let's say, sushi from a freshwater fish is not the best idea you might ever have. Even though here in the United States, pretty much all of the fish which is commercially sold, not the one which you caught yourself, but the one which is commercially sold will go through the flash freezing in order to kill any kind of parasites. If you caught it by yourself, let's say you caught a catfish, that's a great fish, really tender meat, everything is good, flesh. But I would not recommend you to make a sushi out of catfish. Or there is a hei in Korean where you're just cutting pieces, or poke, right, in Hawaiian, where you cutting the pieces of fish and just marinate it for a little while with the soy sauce or vinegar or both, and a little bit of spices, and then eating it. That's great for the saltwater fish, no problem whatsoever. With the freshwater fish, you don't really want to end up with any of those worms. And again, if you want to, that's your choice. But again, in this particular case, we might have, or you might have to have some of those particular creatures living. So anyway, use the diagram above. Length, liver flukes live. When the eggs leave the human body, the next host is the mollusk, a snail. And the next stage enters a fish. So as you can see, several hosts, so complex life cycle. And in this particular case, in order to overcome the odds, a very low odds that particular egg will end up with that particular snail, they have to produce tons, millions of eggs. All right, when the human eats the host, the next stage ends up in the bile ducts of the liver, usually hermaphroditic, containing both male and female structures. Why it's particular useful strategy for a parasite? Well, there is 2 points to that, right? First of all, fast reproduction. Well, you don't have to look for a partner, because it might be just the only worm in the liver. In order to reproduce, have to have a partner, so it can do this self-fertilization first, and second of all, it can produce tons, millions upon millions of eggs by itself, completely fertilized without any kind of. Now let's take a look on that one. As you can see, the structure looks very similar to what we see in the free living, but now there is a few changes. First of all, the gastrovascular cavity drastically reduced in sizes. Why? Because it does not have to digest no more. It feeds straight from the blood. But already have quite a few, quite a lot, I should say, quite a lot of nutrients, right? So those red channels now going through the whole entire body, but this is the gastric cavity now, because once again, the amount of digestion is drastically reduced. They are already absorbing or sucking whatever is has a lot of nutrients. Now we have the blue part, which is right here, and that would most likely be testes. And then this is the, I think we have, yes. So those ones, the blue ones are testes, and the brown ones most likely will be ovaries. So testes produce sperm, sperm goes into the oviduct and fertilizing. All of that are the eggs located right next to the digestive. So all of that you can kind of see tiny little dots around. Let's see right there. Yeah, all of that are eggs. So again, a single worm producing millions upon millions of eggs, self-fertilizing. So ovaries, testes, yes, okay, and those ones are the gastrovascular cavity. So here we go. Ovaries, the brown lines, they are majority because they're producing tons of eggs. Testes, the blue ones, producing fertilization. And gastrovascular cavity now reduced to only 2 channels. All right, table 3. So again, we have some of the, and this is some of how it looks like. That's about the size, about the inch long. This is how you will see liver fluke. And this is the, this is a different fluke, but you can clearly see that's the actual preserved specimen. And you can see the gastrovascular cavity, 2 channels, and then the sperm or the testes and then the ovaries. And here you can pretty much see the same thing. So yeah, that one is a good picture of the gastrovascular cavity. And those pretty the dark spots, that's the suckers. They suck in and stay attached to the liver to get so they will not move. And you can actually see right here, you can see quite a few eggs already being formed in this preserved specimen. Yeah, you can see tons of eggs. All of that is filled up with eggs. So yeah, here we have the life cycle. Now table number 3, we are talking about tapeworms. Those are growing big, but in this case, look, again, we have the final host and temporary host, and usually with the final host, they live in the digestive system, the temporary host somewhere in the blood-rich organs quite often. So let's kind of take a look, and we talk about the echinococcus, I think. No, in this particular case, yeah, tapeworm. The humans can be the final host, and in this case, we're looking at the first as a dog and a cat producing eggs. That has to end up in the water. From water, it has to get into a flea. Then the flea is jumping on the creature, and dogs and cats trying to capture the flea, killing it. Whenever they're killing it, they will swallow it, and then it will trigger the development of the larvae in the digestive tract. So if by any chance you will swallow this thing, it might end up inside of you as well. So yeah, don't eat fleas. Not I'm saying you are, but still. So name insect vector. Vector is what brings that particular parasite toward the final host. And in this case, the vector is flea. Heartworms, the vector is the mosquito. How can a tapeworm? Well, accidentally swallowing the infected flea. Tapeworm body highly specialized, but do not have a digestive system. Why not? Because they live in a digestive tract of the human, or final host. The final host does does the digestion for them. All they need to do is absorb digested materials. All right, makes sense? So now let's take a look at the slides. Well, let's kind of start with the slides. So here we have the slides. Let me kind of move it to the side. So here we have the head of that right here, this part. That's known as the scolex. Yep, right here, the head end. And then it starts creating these small, tiny little bitty segments as the tape form progressing, the segments increasing in size. They start having the ovaries and testes. You can see that the sperm will produce, will come out and will be going into oviduct, fertilizing the eggs. And now all of that, that's a mature proglidid filled up with the eggs. The sperm no longer necessary because it's already fertilized pretty much all of the eggs. So that particular pouch known as the mature proglidid containing tons of eggs, and then it can detach from the main body of the worm, getting into the feces, hopefully to end up in the water. So in this particular case, yes, head. Now we're looking at the, let me see if we have, here we go. You can clearly see the head. This is the suckers to attach to the walls of the intestine because the environment is very slippery. And here you can see the hooks. I don't know if it's eggs actually. Not the creature. Oops. On that one, you can kind of see the hooks. The black thing is coming out, and this is the suckers. So suckers and the hook. What function do they serve? To attach to the walls of intestine, not slide out. Now, whenever you're looking at the mature proglidid, which is this, that part, you can see the sperm coming through, well, the testes producing sperm comes right here and enters the same. So it's not only the same worm can fertilize itself, same segment generally will fertilize itself. So the sperm comes out, enters the oviduct, and then it will go and fertilize the eggs. And after the fertilization of the egg, you will see the gravid proglidid, and all of those dots which you see is actually eggs. One of those particular pouches can have hundreds, thousands of eggs. That the mature worm laying down millions of eggs a day. What type of proglidid will break away and travel through the body? That would be a gravid proglidid. So here we're looking at the preserved one, and here's the head. That one has only, well, scolex, head of scolex. Here we go. Under a higher magnification, you can see that that one has only suckers. It does not have hooks. And then you'll see the body segments. That's mature or gravid proglidid. So now on the model. So here we're looking at the two different scolexes, different species. Here we have hooks and the suckers. That one just the suckers. And then you're looking at the zygote. So zygote with the embryo developing inside. That's the. So here we're looking at the different segments of proglidid. In this particular proglidid, the testis producing sperm. Sperm comes out and goes into that particular oviduct, which has tons of eggs. Now, over a period of time, you can see that the eggs getting larger, bigger, increasing in size, and now the sperm is losing, or the testes are no longer important. They are not developing, but the mature of gravid proglidid, tons and tons of zygotes, eggs. All right. So we have scolex, hooks, suckers, mature proglidid. So here we have a mature proglidid, which essentially has both the testes as well as the ovaries, and a gravid troglodyte will contain mostly ovaries with the zygotes, and that would be the zygote right here. Again, a model of these colics, you can see the suckers and then the hooks. And this is some of the examples of the different tapeworms. Diploidium is not a very large one. Montanessa is the longer one, about 10 inches. So that one can grow up to a couple feet long. All right. Yeah, here we already talk about it. And this is the eggs ready to be hatched out, right? Developing, well, technically not the eggs, but the larvae. Now, trichinosis, as you know, that in many cultures that the pork considered to be a dirty animal, or swine considered to be dirty animals and not good to eat. And that's probably one of the reasons why our ancestors says that pork, because once again, quite often the porks will have the parasites which have no problem to go and parasite, well, to become a parasite of humans. Physiologically, we are very similar. So, and yeah, one of the tapeworms can do that. So now nematodes, right? Nematodes, roundworms, bilateral symmetry. Now with complete digestive system with the mouth and anus, and then pseudocelamates. And remember, they shed, well, excellent. Now looking at the anatomy of male versus female, let's take a look right here. This is something which is known as a horse hairs. That's a free living, it's still not really a free living things they do, but the larvae parasitize on insects. So in this particular case, you'll see the worm, right? And then you will see the worm, and then there is a hook outside. Which one is a male? The smaller one with the hook, same thing. This is Ascaris. This is a human Ascaris, I think. So in this case, the female are much larger. It was so long that they had to bend it, but the male is smaller, thinner, and again has a hook to literally the female. Yes, we are talking about figure of speech, but in this case, hook up with a partner, in this case female. So again, that's a male for sure, that's a female for sure. You don't have to worry much about the anatomy. It's really hard to see, well, the intestines, it's all marked, but don't worry much about that. You can take a picture of that, that's pretty much it. But be able to recognize male versus a female. Now, some of the other representative of the roundworms are vinegar eel, that particular microscopic photograph of eel. And vinegar eels are the creatures which lives in the rotting organic materials. They're usually trashing around, and I think there is a question asking why they are doing the trashing material. Basically, whenever they trash all around the liquid, they're trying to get any kind of small food particles into the mouth. And then here we have some of the heart with the worm inside. So if you'll see something like that, and it will say name the creature, don't say dog heart, no, talking about the worms. So yeah, let's see trichinella. Here we go. Here's a trichinella. So this is the muscle, essentially, pork chop right there. And in some of them, some of the infected ones, you might see those particular structures. So all of that muscles, blood vessels, and those are the larvae waiting to hatch. Okay, so you have muscle, and this is the cyst. Several of those. This is another cyst, and I think this is the larvae right here. You can see kind of like a worm, and this is the worm as well, right here. So yeah, consuming that, I mean, if it's a fully cooked, no problem. And you might have heard that you're not eating the pork pink. Pork should not be pink. That's one of the main reasons. Any kind of possible parasites. Because again, physiologically, from the pork, switching from the pork to a human, not a big problem. On another hand, if you are like your steak blood example, right? But steak is the cow meat, right? It's beef. Beef, some of the parasites can go to us, but generally we have a different physiology. They are herbivores, we are omnivores. As a result, the creatures will not really survive. So yeah, observe the vinegar eels. This is how the vinegar eels looks under a microscope. Generally, you have a slide, you dropping, we have the colony of them, and you can see those particular guys, and they are thrashing all over the place. So quite interesting. But they are harmless. They live in a decaying materials. All right, now we're looking at the dog heart with the worms inside. You can see the worm sticking out, and the vector is the mosquito. We talk about that. So, infected mosquito. Not all of the mosquitoes carry that. So now we're moving on to the mollusks, and in the mollusks we'll talk about three major classes: bivalves first, then we'll talk about the gastropods and the cephalopods. So sketch a scallop. Well, let's take a look at scallop, right? And there is a, make sure that you know the labeled things. So number one, what's the number one pointing to? The thing which is surrounding the internal part. That would be a mantle. Now, number two, that muscular organ, that's a foot. Number three is kind of talking about the overall thing known as the visceral mass. Number four and five are abductor muscles, which is closing down the shell. Number six are gills. Gills, then we're going into the intestine, right? And then there is a stomach. And number seven right here, number seven is the heart. So mantle, foot, visceral mass, abductor muscle, gills, and the heart. Here we go. I think it goes in the proper, yeah, it goes in order. It's listed. Now sketch a scallop. So here we go. Make sure that you understand that visceral masses, everything inside is covered by the mantle. So here we have the sketch the scallop. Let's see the here we're looking at the oyster. That's the scallop. That's the shell of the scallop, of course, right? And the sketchy oyster, all you need to do is take a picture of that snippet. Actual oyster, clam, and then the scallop. This is a spiny oyster. That's the abalone, which is a gastropod you can find in the Pacific Northwest. Very tasty. I think I saw it somewhere. The preserved ones you can buy canned abalones, but can was like $50. And this is mussels, typical mussels again. Pacific Northwest has tons of those. You can, whenever we're spending some time in Oregon, we were on the coastline. We went bought the fishing license, and then you can go get them on the shore and then cook them. Really nice too. Anyway, so let's take a look at the gastropods. Remember, those ones are snail and such creatures. So here we're looking at the snail with the shell and then the food. Here we're looking at the lightning whale and the egg pouches. Inside of the egg pouches, you'll see those particular eggs. Why we are talking about a particular gastropod, which is known as lightning whale? What is the importance? The importance of the lightning whale, this is a state shell of Texas. You know, there is a state tree, which is a pecan. There is a state flower, which is a bluebonnet. And the state shell is the lightning whale. All right. So here we're looking at the preserved snail. let's see, scale, shell, foot on the head, shell, foot on the bottom, and then the head. And then sketch this slug. I think I have, here we go. It doesn't, so it has the foot, it has the head, and no shell. Sketch lightning welcome with the egg case. Make sure to be able to. This is a large shell known as the murex, and we talk about the murex, the purple dye. Yes, ancient Phoenicians used secretion to create a beautiful purple dye. Then it moved into the Roman Empire and then to the French Empire. So yeah, extremely expensive. So now that's the gastropods, and in the gastropod, what you'll need to be able to recognize the suckers, the gills, the red ones right here, the gills. That part, the open part, the calamari, actually rings of calamari. What you are eating essentially a mantle. If you are ever eating, this is the siphon, and then we are talking about the tentacles. There is the arms and the tentacles, and it's kind of easy to recognize when you're looking. They usually have 10 arms, and 2 of them will be a much longer. 2 of them. So the tentacle is the usual one, and the arms are the longer ones, and the longer ones are used for the reproduction and the cord shaping. The male grabs the female, and so on and so forth. Pacific, one of the largest octopi in the world is the Pacific octopus. It mates only once in its lifetime. After the female took care of the little ones, it dies. So visceral mass is all that particular stuff. If you ever clean calamari, you're pulling out, so that's you're pulling out all of the junk. That's the visceral mass, and the tentacles and the arms. All right, so here we're looking at the octopus, right? And then this is it, and you can see that 2 of them are much longer. That's the arms. And here we're looking at the Nautilus, the shell, of course, of the Nautilus. This is the shell of Nautilus, which the top layer has been stripped out. As you can see, it's exactly the same. And this is the Nautilus, which has been cut through. And you can see that the first chamber, this is where the Nautilus resides, and then there is those pressure chambers. This is the Nautilus, which is roughly 150 millions of years old. That's a fossil. And again, you see the main chamber and then the pressure chambers. Here we have a wonderful fossil. Our department had bought it, and you can see a nautiloid, an ancient one, and then this is the straight, something which is known as an orthoconic nautiloid. Initially, as far as we understand, they were straight cones, and then it started bending over, creating that circle. So sketch the Nautilus. Take a picture, make sure that you will be to identify. And here we're looking at the arms, right? Yeah. Yeah, 16. Nautilus, and the octopus is right here. That one would be octopus. This one is octopus as well. And here we're looking at the preserved Nautilus. You can see the actual mollusks sticking out, and it will occupy or reside in the largest chamber. So yeah. All right. Next table is number 8, and number 8 talking about the annelids. We have a model, and I'll point it out what exactly needs to be known in the model. Many of those things are kind of hard to identify. So first, mouth, the open end right here, mouth. Then the mouth going into a pharynx, that green part. And then there will be a thicker one known as the crop and gizzard. Don't really see that right here. The overall green passed this way, it's known as the intestine. Don't have to worry about those ones. Now, aortic arches. Aortic arches, remember, those guys have a closed circulatory system. Now, that one, the main blood vessel known as the aorta. from the aorta, you will have the blood vessels coming on either side, creating like an arch, and those ones are pulsating. So essentially that's the hearts, and there is 5 of them on either side. So earthworm technically has 10 hearts because it's on either side, but in this particular case, this is aortic arch, another aortic arch, another aortic arch, another aortic arch. Seminal vesicles, seminal receptacles, ovary, testes, all of those kind of hard to see, most likely will not be on a lab practical. I'll try not to put it in. If I'll do and you will call me out on that, I will give you the full credit. However, clitellum, very interesting name. You see that kind of a thick band right there? That's the clitellum, very important for our sexual reproduction. So whenever 2 worms coming over, well, trying to cross fertilize, they actually will be kind of bending at the clitellums. So yes, that one is that particular band of the known as the clitellum. So here's some of the examples of the annelids, and in this case we're looking at the earthworm, and this part is the clitellum. This is a segmented worms, polyheta, and this is the leech. So yeah, you can kind of see, but it's very small, very hard to identify. So, but your mouth, that's for sure. You should be able to identify the green part known as the intestine, aorta, the very large blood vessel, aortic arch is coming out, clitellum. Pretty much all you need to know for the lab lab practical. And again, you can see the mouth right here. Yeah, here I removed some of the pieces. Yeah, I removed the in this I see the ovaries and the testes, but you don't have to worry about that. I will stay away from those particular photographs. So that's my promise to you all. And again, we're looking at the preserved leeches here in there, and this is quite often they will create the intricate pathways, or essentially the boring place on the top of the shells. So yeah, polyhead, you can clam worm, you can see tons of those particular bristles. And in this particular case, this is the head, and you see those bristles increasing in size because they start helping with the breathing. So, and I think that's pretty much it for the lab. Post-review questions. Which animals first to have cephalization worms? Name only cephalopods with heads external shell. That would be an Nautilus. Scallops belongs to adults. Trichinella belongs to the roundworms. How can you differentiate between male and female? What organism is a descent of Nautiloid? We talk about that in the lab, or in the lecture. I briefly mentioned it, known as the Ammanoid. Let me type it in. So we're a much more successful, much larger group, but about 65 million of years ago, they died off along with the dinosaurs. They derive from which organism? That would be a murex. Name the vector which transmit heartworms. Mosquito. Name the organs that increase soil fertility. A member of which phylum? Earthworm phylum Annelida. You can see it right here. Name a member of gastropod. Snail. Slug. Name a member of the cephalopod. Squid, as well as the octopus, as well as the nautiloid. Marine worms belong to what phylum? Most of the marine worms will belong to the polyheta, many bristle creatures. All right, now let's take a look. Characteristics of three major phyla of worms plus the mollusks. Platyhelminthes, type of symmetry, bilateral. As a matter of fact, you can put bi in each one of the particular thing. So bilateral, 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 bilateral. They all bilateral. Number of germ layers. They're not cnidarians, so they all triploblastic creatures. All triploblastic. Now, Platyhelminthes, asolemate. Nematoda, pseudosolemate. Annelida, as well as the mollusk, are true solemate or eusolemate. You can just put solemate. Incomplete digestive tract for the flatworms. Everybody else have a complete digestive tract, the beginning and the end, the tube now. Segmentation, no segmentation in the flatworms as well as the nematodes. Annelids have the segmentation. Mollusks technically do not have segmentation. Circulatory system, Platyhelminthes absent. Nematoda, if they have it, it's open. Annelida, they do have it closed. Mollusks present, most of them open except the cephalopods. Cephalopods has a closed. Now, phylum, cladogram phylum. What do you need to do? You need to add the arrows. So bilateral, right? Bilateral will split into two: deuterostomia, deuterostomes, and the protostomes. And deuterostomes we'll talk later on. We are talking about the protostomes now. The protostomes split into two: lochophores and dicosaur. So splitting into two. Dicosaur, we have nematoda and arthropods. In Olochatrophozoa, the Platyhelminthes, mollusks, and very, very simple. As a matter of fact, you have that particular table right here, kinda. Look right here. We're talking about the bilateral splitting into protostomes and deuterostomes. Deuterostomes we are talking about later. Protostomes will split into two: Lochatrophophore, Exitosaur. So basically flip. Same thing. Phylum Nematoda, phylum Arthropoda, Annelida, mollusks. Same, right? Just flipped over. So whenever you will be filling out, I would like to have actually those particular errors. And it's relatively easy to do, right? You're copying it. Oops. So yeah. So here we have one, and then you grab another arrow, putting it into that, and then that into that. Don't just do something like this. Don't. Okay. Don't. I had students doing this before. No, no. Don't do that. Some of the students went a little bit further. They took this and they start. They were they were doing something like that. Yeah, no, no, please don't do that either. You can use this particular table as your reference where those arrows supposed to be going, right? But other than that, easy. You'll need to put 1, 2, 3, 4, 5, 6, 7, 9 arrows. That's all I'm asking. All right, main purpose, what you observed, how you can apply. Yeah, well, we talk about a lot of things, right? Including what you want to eat, what you don't want to eat, and so on. So, and we are pretty much done for today. Thank you very much. I'll see you